Okay, final check here. Jessica, can you hear my voice? And can you see the screen?
Can indeed.
All right, sounds good. We will give it, till 10:05 and then we'll start, but I can just start by saying thanks everybody for joining. You are here for Seriously the webinar. I hope you all are doing well this morning and staying safe. So, just gonna wait. For those of you that are astute observers, I am giving this talk from the comfort of my garage office, as you can see from the amount of stuff piled behind me. Okay, 10:05 a.m. I'm going to get started. Hi, everybody. My name is Chris Troll, and I'm the Director of Assay Research and Development at Claret Bioscience. Joining me today is my colleague, Dr. Jessica Morgan Garcia. She is the Chief Production Scientist for Seriously. I will be presenting this webinar. She will be in the background answering questions. So feel free to type questions into the question and answer box as we go along in this presentation. She will answer them as they come up, and then any questions that are salient or she needs further explanation on, we will answer those at the end of the talk. So just to get started, a good starting point or a good additional resource for those people that wish to look into this further is our methodology article that we published from in BMC Genomics at I believe it was at the beginning of last year. And it's an open source article. It details a lot of what Seriously does and how it does it, and gives a very brief methods on it, which we have expanded upon further when we have launched the kit. And for additional resources, you could always go to our website, www.claretbio.com. So to start this webinar, I'd like to orient everybody in terms of just Illumina Library Prep 101, Next Generation Sequencing 101, at which point I'll get into Seriously the method, some of its applications, and then some of its fields of uses and what we currently offer. So to orient everybody in terms of what a complete Illumina sequencing library looks like, this is it. Your insert is in the middle, and then upstream of it, there is your P5 adapter, which is in all essences your read1 sequencing primer, as well as your I5 index primer as well, if you depend on using dual indexes. And then above that, you have your bridge amplification and flow cell binding portions. Downstream of your insert, there is your P7 adapter, which is also your read2 sequencing primer and your I7 index primer as well. Then you have your I7 index and then your bridge amplification and flow cell binding stuff. Regardless of how any next generation sequencing libraries get made, this is the core of what they look like after index PCR. Both the upstream and downstream parts to your insert or your template DNA is both about 65 base pairs of dual DNA, of duplex DNA. So the adapter dimer itself, when there is no insert present, is 130 base pairs. So there are two basic ligation-based approaches in order to get your adapters and the entire Illumina portions of your DNA onto your insert. The first is the stubby adapter approach. And the stubby adapter approach is when you ligate on short adapters. And these are basically just the sequencing primers or the adapters. They're about 30 to 35 base pairs long. And then the indexes and the bridge amplification portion of the adapters are added during the index PCR step with complementary parts to part of the P7 for the round 1, and then complementary parts to the P5 for round 2. And both these continue on through rounds 2 through N, however many cycles of index PCR that you choose to do. In contrast to that, the second ligation-based approach is the full-length adapter ligation approach, where basically the entire sequence of the adapter, the index, and the bridge amplification portions are added during adapter ligation, and then subsequently through index PCR, the primers bind downstream and upstream of the indexes and amplify the whole molecule. There are pluses and minuses to both methods of this. None is wrong, both are good. But for the purposes of what SRSLY is, SRSLY adapters are, in essence, these stubby adapters. That is the approach that we take with SRSLY, where we first ligate on the short adapters, and then we index and put on our indexes and our bridge amplification primers. The main way in which SRSLY distinguishes itself from, say, double-stranded preps is how these sequences get onto the insert, because the template DNA needs to be prepared to get adapters ligated onto it. So I'd like to take a few minutes to just kind of discuss how that occurs and the differences between double-stranded and single-stranded preps. So so your template DNA is usually a diverse pool of DNA. It is never a homogeneous pool of molecules. Depending on the sample type, it is more of one type than another. Shared genomic DNA, which is high quality DNA, will have a large majority of double-stranded molecules with blunts and overhanging ends. Degraded DNA, such as FFPE, cell-free DNA, ancient DNA, will have some of these molecules, but also a higher proportion of single-stranded DNA and nicked double-stranded DNA and short fragments. cDNA from RNA-seq will be primarily, actually exclusively, single-stranded DNA. So the pool depends on your input template type. But in order to prepare this template for double-stranded library preps, the first thing that happens is end polishing. And during end polishing, T4 DNA polymerase blunts all double-stranded molecules by chewing back 3' overhangs and filling in 5' overhangs. I'm going to reverse and go back into this to show you the little animation I have done because it's kind of important. So blunt molecule stays the same. So if you focus on the 3' overhang, these molecules get chewed back to blunt due to the exonuclease activity of the T4 polymerase. And then if I back up one more time and look at a molecule with a 5' overhang, it gets filled in. So after that, a modified Taq polymerase comes in and it puts on 3'A tails, which facilitates TA ligation to your double-stranded adapters, allowing to use a slight overhang to make the adapter ligation with T4 ligase more efficient. With the double-stranded adapter ligation comes some caveats. And the first is the double-stranded adapter ligation, the adapters themselves can ligate to double-stranded DNA. They cannot ligate to single-stranded DNA because they don't have the capability to do that with requiring double-stranded overhangs and a double-stranded complete molecule to make your library molecule. And in essence, you'll lose the top strand. You'll lose a nicked DNA strand, not so much for the adapter ligation, but downstream when these two molecules disperse to go into index PCR. I'm going to back up and show that again, that the top strand of the nicked molecule disappears as well as the double-stranded molecule. This is the general structure of a double-stranded adapter, where it's a Y adapter that's got a P7 and a P5 prime, and then a T tail here to complement the A tail in the overhang. You can see that a nice end polished and A-tailed double-stranded molecule fits in here to ligate very nicely, but you can envision that a single-stranded molecule does not. Neither does if a nicked template is here with a missing a basic site or just a single-stranded break, that this portion will denature from this portion during index PCR. Due to size selection stringencies, you lose short fragments. This can be overcome depending on the SPRY purification you use or the bead purification you use, but generally double-stranded preps are geared to not retain as many short fragments as single-stranded preps. And that could be there's a few different theories of whether the single-stranded DNA is a major component of the short fraction or the nicked. So that's a double-stranded prep in essence. A double-stranded ligation-based prep is how it works. So Next, I want to show a slight animation on how a single-stranded DNA prep works, seriously. So again, you have your diverse pool. I'm only showing a subset of the molecules I showed back there because there is no end repair step in seriously. So regardless of the double-stranded molecule, whether it's blunt, has a 3', 5' overhang, it doesn't matter. These are the three molecules that you want to look at: double-stranded, nicked, and single-stranded. So the very first step in seriously is an initial denaturing. So all DNA molecules become single-stranded regardless of their starting conformation. So double-stranded duplex molecules will become two single-stranded molecules. A nicked molecule will actually become three molecules because this nick will turn into a single-stranded break, and a single-stranded molecule will remain the same. After that, there's a splint adapter ligation step, and the splint adapters look like this in this crude animation. In the next slide, I will show you a better figure of how this works. But basically, there's double-stranded portions of the P5 and P7 adapter, and the splint is on the bottom, and there's random bases here, which will then nestle nicely into complementariness with your template DNA that has become single-stranded, and this gives it a localized double-stranded interaction to allow T4 ligase to do its work and put on these adapters and then go through index PCR. If I can get 1 point across to everyone here in this seminar, it would be that is this: regardless of the starting state, all DNA molecules are ligated as single-stranded DNA. That makes this a single-stranded DNA library prep. There is a misnomer in the field that single-stranded library preps only capture single-stranded template molecules. And that is true in that all template molecules are rendered single-stranded, but untrue in the fact that it doesn't capture molecules that were double-stranded. So single-stranded DNA molecules in single-stranded DNA library preps, in theory, can capture a broader diversity of input molecules than double-stranded DNA preps, based on this initial denaturing and the fact that single-stranded molecules and nicked molecules can be captured. So the standard workflow for SRSLY follows. So the DNA input molecules, this is somewhat of a rehash of the last slide, but a diverse pool of inputs. Depending on your input type, it'll be more skewed towards damaged and single-stranded versus fragmented and double-stranded, which is high quality versus kind of lower quality DNA. There's initial denaturing step. We add in a single-strand, a thermostable single-stranded binding protein to capture the single-stranded molecules and keep them single-stranded. A cold snap after the heat denaturing also helps this. What you show here is the nicked molecule going into three molecules. And then you have your splint adapter ligation, where the forward adapter here is double-stranded until you have a single-stranded portion of the splint that's 7 bases. Same thing, but opposite for the reverse. Forward is your P5 adapter, reverse is your P7 adapter. 7 random ends here to create the localized double-stranded ligation. The total time of the prep is slightly shorter than a traditional ligation-based double-stranded prep, and that's because there is no end repair step in SRSLY. The only part of SRSLY that we do for end repair is we phosphorylate the 5' end and dephosphorylate the 3' end if the molecule is not already in that conformation, and that happens during the same time as ligation. So it's a 10-minute denaturing step, then an hour ligation phosphorylation step, then a cleanup, and then index PCR for 30 minutes, and then cleanup. At the end of SRSLY, you get a standard Illumina sequencing molecule. And if we zoom in on just the important part of the molecule, not to say the bridge amplification part of the molecule isn't important. It is, but it's ubiquitous across all molecules. But the SRSLY molecule itself looks like a standard one, and now we've included in our catalog offering these 8 base pair UDI indexes for your dual indexes for your I5 and I7 index. UDI meaning unique dual index. And the only reason I'm making a point of this to say that this is now what is included in the SRSLY kit when you buy it is because during our initial launch, we did combinatorial indexes included into the kit, and the combinatorial indexes basically means one index on either the I5 end or the I7 end can be used multiple times with different indexes on the other ends to create multiple libraries. While this works and is efficient and is generally cost efficient, it has issues when it comes to newer patterned flow cells, such as the new next generations of the HiSeqs as well as the NovaSeq. So because there are index hopping issues, we've done away with our combinatorial indexes, and now we offer unique dual indexes for our kits. And that basically just means an index is never repeated. An index barcode, an 8 base pair barcode, is never repeated on either the I5 or the I7 index. So as you can see here from index 1, 2 through index N, these are offered in 12 and 20 and 96 reaction volumes for a kit for those of you who are interested. So next, I want to talk about the UMI workflow of SRSLY because we have a way to incorporate UMIs into your library if you are at all interested. UMI meaning unique molecular identifier. These are random bases that are inserted somewhere into the sequencing library to count unique molecules. It is especially important and used heavily when people are doing rare variant callings from cancers, cell-free DNA, FFPE stuff, people who are interested in extreme levels of complexity from very low complex samples such as oligos or amplicon libraries as well. This is the applications in which you want to use UMIs. So the UMI workflow is somewhat elegant in that the base reactions still occur. The denaturing, the adapter ligation is all the same, but then before index PCR, a linear amplification step is added that amplifies, it doesn't amplify, sorry, it extends and creates a complementary strand to the ligated template strand that includes a UMI. I'm going to enlarge this figure here in the next slide to show you exactly what I mean. So after SRSLY adapter ligation, you have your P5 and your P7 adapters ligated on there. And then you denature again and you re-anneal to a primer molecule here that is complementary to your P7 adapter. It has your I7 index in there and it has a 9 base pair random UMI nestled in between the I7 index and the flow cell binding part. This linear amplifies in this step and goes all the way across here. Then during index PCR, different primers are used, and the primers that are used are ones that put on your I5 index. I misspoke. In essence, your I5 primer is the same, but your I7 primer is different in that it becomes a full-length adapter primer, if you remember from the beginning slides, where it basically just binds to the flow cell section and truly copies both the UMI and the I7 index that is added in this step. At the end of the UMI protocol, what you get is a molecule that looks exactly the same as the initial molecule, except for this little UMI that's here. And it's tagged, in between the I7 index and the bridge amplification portion. So UMI is 9 base pairs and random. It is it's a unique molecule. There aren't many that are created like this, and it can be tricky to extract this information and to tease apart what is I7 index and what is UMI. So in order to make this easier for our customers and anybody else really who uses this sort of scheme to put on their UMI, is we have made some free software available that's at claretbio.com software, and it's SRSLY UMI or SRSLY Yummy, depending on how you want to pronounce it. And what it basically does is it takes your BCL files and your runinfo.xml sheet, and it outputs FASTQs that are parsed correctly. And I'll show you kind of how it does this. So the base calling cycling order that So this is construct, but the way the machine reads this is it goes read 1, read 2. This is included in your read 2, both the I7 index, your UMI, read 3, and then read 4. And so with the run info.xml, the software will relabel this as 1, 2, 3, 4, 5. And then if we go to the next slide, what happens is in your output of your FASTQ, once you put this through your seriously yummy software, is you will get two FASTQ files out of this, your read1 FASTQ and your read2 FASTQ. And within the header row of the FASTQ file, because FASTQ files are 4 lines, you will get your at flow cell coordinates. Then you will get the UMI sequence. You'll get the 9 base pairs. And then just for confirmation, you'll get your I7 index, which is number 2, plus your I5 index, which is number 4 in there. Then you'll have your sequence. Then you'll have this plus or a repeat of the flow cell coordinates, and then your quality score for your FASTQ. So this hopefully stems a lot of bioinformatic issues that people would have coming from this. And then from there, customers are free to do their own analyses, which use the UMI. So SRSLY has 3 main pros, 3 main highlights of it that separate itself from your standard double-strand library prep. And that is that it captures a high amount of single-stranded and nicked DNA inputs, higher amount than double-stranded preps, higher in that it captures more than 0 than single-stranded inputs and more than 0 nicked ones. It has native termini retention at both the 5' and 3' end because there's no T4 polymerase in the SRSLY ligation reaction. So the 5' end remains native for what it's worth in both double-stranded and single-stranded preps, but the 3' end is altered due to the T4 polymerase either chewing back or filling in. SRSLY doesn't alter the 3' termini. And it retains a higher amount of short fragments. This is something that has been demonstrated multiple times over in many different single-stranded prep methods that have been published. So I want to talk for a minute about cell-free DNA because that is the kind of flagship template that we market SRSLY for. So cell-free DNA falls right here in terms of the Venn diagram. Native termini retention is important for cell-free DNA, and short fragments are important for cell-free DNA. And I will demonstrate why that is right now using some of kind of the current literature for those people that aren't super familiar with cell-free DNA biology. So a quick background on cell-free DNA. For what it's worth, cell-free DNA derives from apoptotic and necrotic cells that are either located in the blood plasma, urine. What else is important? Where else is cell-free DNA found? It is found in the aqueous humor. It is found in cerebral spinal fluid. For the purposes of this talk, I'm talking about blood plasma cell-free DNA. Each has their own kind of semi-unique features, but blood plasma DNA is far and away the most popular way in which to capture cell-free DNA. Within blood plasma cell-free DNA, the majority of DNA that's in there is from apoptotic myeloid and lymph cells, but there is a very tiny portion or large portion, depending on your clinical state, of either cancerous cells, fetal cells, or organ donor cells. These are the 3 main fields in which cell-free DNA is being applied as a liquid biopsy to look at cancer progression, to look at kind of fetal health, neonatal health, and then organ transplant or rejection as well. Cell-free DNA is naturally very short. If you look at an insert length profile of cell-free DNA, this is what it looks like when it's extracted somewhat properly. There may be a second peak here that's a dinucleosome, but far and away it has a huge peak right here at about 165 base pairs, and this is a histone with the linkers attached, actually. And this is because once a cell dies and opens up, there are DNases that attack the DNA and chew back and basically degrade the DNA, but DNA that's wrapped around a nucleosome is more protected than DNA that is naked. So that is why you get this huge peak here. Then there's basically a 10.4 base pair periodicity, because this is where DNase can nick the DNA as it's wrapped around the histone in the minor groove of it. So that's why you get peaks here and here and here as the DNA that's protected by the histone is slowly degraded over time. And so all of these nicked molecules represent DNA that has come from histone protected DNA, but there is also a subfraction of DNA here that is enriched for other interesting biological information. And that's kind of what I want to talk about. And this is what I mean by seriously for short fragment retention in relation to seriously for cell-free DNA applications. So there's been a growing body of literature that's shown that the short fragments of cell-free DNA are enriched for additional information other than nucleosome-bound DNA. There's a nice paper out of University of Washington in 2016 that showed that these short fragments are enriched for transcription factor-bound DNA. So in addition to DNA, long DNA being protected, relatively long DNA, 165 base pair DNA, being protected by wrapping around the nucleosome, there is DNA that is wrapped and close enough to transcription factors that are much smaller than nucleosomes, and that those are retained in the short fragments as well. A paper out of Cornell recently showed that the short fragments are enriched for microbial and mitochondrial DNA as well, for people interested in looking at kind of microbial or mitochondrial phenotypes from cell-free DNA. It has been known since the birth of kind of the cell-free DNA, well, what gave birth to the cell-free DNA application for neonatal health is the observation that fetal DNA fragments are shorter than maternal DNA fragments in cell-free DNA. A lot of this information has come out of Dennis Lowe's lab from University of Hong Kong, who's pioneering fragmentomics, which I'll get into in a few minutes here. And then a nice paper out of Johns Hopkins that came out recently showing that the ratio between short and long fragments is dysregulated in cell-free DNA of patients with cancer. And this can be a way of looking at the cancer progression in people with, in this study with lung cancer. As you can see, aberrations between the mean at 0 in this lung cancer versus the healthy cell-free DNA. So that is the rationale for why short fragments are important in cell-free DNA. And here is a length distribution profile of using seriously versus a double-stranded DNA prep, showing that seriously, when applied correctly with its short fragment retention policy, can capture a majority of short fragments in this subnucleosome level where they're enriched for. And that's what you see with this red line here over the double-stranded prep, which is orange. This is a phenomenon that is not unique to seriously. It has been seen in several other single-stranded preps. And if I back up one slide, if you look at the University of Washington article or this Scientific Reports article out of Cornell, both of those show similar figures to what seriously has here and recapitulate the point that single-stranded preps do indeed capture a higher proportion of short fragments when applied with cell-free DNA templates. The second selling point for seriously in terms of cell-free DNA applications is its ability to retain native termini retention. So all of these articles that I'm going to show in succession here come out of Dennis Lowe's lab in University of Hong Kong, who has been instrumental in this fragmentomics approach to cell-free DNA. Fragmentomics meaning this is a biologically fragmented molecule as opposed to genomic DNA, which is either acoustically sheared or enzymatically sheared to make it short. This is naturally short, and it goes through a degradation process to do this. And by studying how this happens and what the fragments look like, you can learn stuff about the history of it or where it came from or where it's going. So a recent paper out of Dennis Lowe's lab shows that there are preferential ends in age cell-free DNA, depending on whether it comes from whether it's being degraded. If it's early on in its lifespan, then it's got some preferential ends. If it's later on in its lifespan, then it has other preferential ends, depending on what DNase is acting on it at the time. There was another paper that shows that plasma DNA from people with chronic liver cancer, that's this HCC, they have a preferred CCCA end motif in their DNA, and this can be used as an additional fragmentomic biomarker when looking at hepatocellular cancers, in addition to kind of the standard mutational profile. And then finally, maternal plasma and neonatal, prenatal plasma, there are different preferred end profiles in those as well. So that is the rationale for why native termini are important for cell-free DNA and how researchers can further that field in using SRSLY for this. This is a figure from our manuscript that kind of demonstrates that SRSLY has native fragment retention. The point of this figure is to show that if you look at the double-stranded prep, there's a lot going on here. So just to simplify it, the ends of the molecules are at 0 here for the forward and reverse reads, and then it reads into the read this way to orient yourself. As I mentioned before, DNA, cell-free DNA is degraded biologically, and there is non-randomness to its ends. So that's why the ends look so much, have a stronger preference than as you get into the molecule in terms of its normalized proportions of bases to the human genome. As you get into it, it just looks like the human genome, but at the end it's frayed and different because of all the different biological important factors that are going on. So that's, and then the bases are colored, T, C, G, A. And the whole point of this figure is to show you that the end here for this double-stranded prep is exactly the reverse complement of the end here for the forward and reverse read. So what is blue here, what is C here, is G here. What is T here in red is A here in green. And this is due to the T4 polymerase making the molecule blunt before the adapters are ligated on. So every read is the reverse complement of each other by the fact that all molecules are blunt and double-stranded. That is not the case for SRSLY. Here you see the same profile as the double-stranded prep for SRSLY because the 5' end is left unaltered, regardless of whether the prep is double-stranded or SRSLY. And then the difference here is that the 3' end is not the same as this 5' end, and it's not the same as this 3' end, indicating there is no end repair here, and this is a different profile than you get if you end repair. Some other quick metrics just to go through for SRSLY for cell-free DNA. This is just to show that SRSLY passes the sniff test in terms of the library molecules look as they should. SRSLY shows the GC percentage across the base percentage of the human genome here, as you would expect. SRSLY is in red and pink, and the double-stranded prep comparison is in black and gray. It is worth noting that SRSLY is slightly higher in GC percentage. This is probably true in relation because cell-free DNA has a higher proportion of CpG islands than normal background human genome here, and that is due to biological reasons, which I won't get into. The second thing, the second sniff test metrics show that the fold coverage for SRSLY across the percent bases is just a normal Poisson distribution, which is what you'd expect in an unbiased kind of library. You don't want to see some bimodal distribution where some parts of the genome are getting overly covered than others. So SRSLY has that, and so does double-strand prep. Complexity, this is just the expected unique reads based on the duplication rate at the levels these libraries were sequenced. SRSLY has a very good complexity here as well, with this gray line on the XY axis showing infinite complexity, which is never the case. Going out to 300 million reads, in terms of total reads sequence, because that's about the high-seq which you would get back. So while I've shown you some parts of some applications for SRSLY and some of its features for cell-free DNA for fragmentomics, there is a very real and tangible field for cell-free DNA, and that's mutation calling within the cell-free DNA to correlate that with cancer and tumor kind of development and metastasis. Mutation and SNP calling and mutations are still king. Fragmentomics is trailing behind. Hopefully it won't trail behind for long, but mutation calling is still the stock standard way in which cell-free DNA applications are being applied in the clinic. And it will be for some time, and it will be for FFP as well as tumor biopsies as well. I mean solid biopsies, not liquid biopsies. So SRSLY works well for that. I'm not going to go into this right now, but we've published a really nice application note on combining SRSLY with the Twist Biosciences exome panel in order to look at CT meaning circulating tumor DNA, SNP profiling. And what you can see here is just a example figure from our app note showing the correlation between in this app note, we had access to both blood plasma and primary tumor biopsy from patients. So you can see the correlation between the variant alleles frequencies that were called within the lung cancer biopsies from that called within the cell-free DNA. And then the impact mutation spectrum based on kind of the potential impact that this SNP has there. But look for more on this on our literature page, and we will have another app note coming out soon that is very similar to this, but using Twist Pan-Cancer panel, which is a much smaller panel than their exome panel, and using our UMI applications to look at very low frequency SNPs that we're finding in several cancer samples. So something to look forward to. The second part of SRSLY that I want to talk to real quick is its applications for RNA-seq. RNA-seq is a huge market. It is about 70% of all sequencers runs are on RNA-seq data. RNA-seq is by default a single-stranded input, and it can or cannot have short fragment retention depending on if you're interested in microRNAs or not. The main strength of SRSLY for RNA-seq is that you don't have to do second strand synthesis. Traditional double-stranded DNA RNA-seq preps require double-stranded second strand synthesis in order to ligate the double-stranded adapters on there. Then you have to remove the strand for directionality. This is usually included into kind of the adapter prep step as well. So this doesn't save that much time, but SRSLY does save time in that it doesn't have any second strand synthesis, and therefore there's really no issue of directionality in your library prep because it is directional by default, because SRSLY just ligates to that first strand cDNA. So it's a much shorter library prep and has the potential to be much less problematic. Some very simple kind of general library metric slides for RNA-seq showing that comparing SRSLY to double-stranded preps, the correct strandedness is there, and the uniquely mapped reads is very high when applied to in this case, this is actually an mRNA-enriched RNA pool as opposed to kind of ribosomal depletion. But ribosomal, very low, unmapped, very low for all of these. Both these preps look fantastic. SRSLY just was a shorter prep. Same thing with these general library metrics. SRSLY and double-stranded DNA preps have similar gene body coverage across the transcripts, and as well as a similar distribution in terms of its read data for here in both intergenic, intronic, untranslated regions, and coding regions. I'm not going to spend a lot of time on this because the whole point of this is just to show that the red bars and the black bars are very similar across the preps. What I do want to spend a few minutes talking about, I am going to speed up because I'm running a little bit behind, is that we've done a SARS-CoV-2 enrichment experiment in conjunction with Arbor Bioscience using SRSLY for RNA-seq preps, as is very fashionable with the time, unfortunately. But this is a proof of principle experiment showing that what SRSLY is capable of for RNA-seq when combined with enrichment. The setup for the experiment is very simple. It is host plus viral RNA at different amounts going through SRSLY, and then a SARS-CoV-2 enrichment panel. The way this was done is into 50 picograms of human RNA. Titration's amount of viral copies of SARS-CoV-2 were spiked in from 1 million copies down to 0 copies. At the 1 million copies, this is 15 picograms of viral RNA. So 15 picograms into 50 nanograms at the highest ratio. Here, this is in the femtograms area. I believe that's below picograms. So, and then pre-enrichment sequence, post-enrichment sequence, fold enrichment. None of this, all of these metrics are very nice to look at, and you can spend time looking at those in our app note. But this is kind of the punchline of the paper and the app note. So on the x-axis here, you've got the viral copies from high to low, and then you have the percent SARS-CoV-2 genome covered at minimum of 1x at different read depths after enrichment. And what you see here basically is that when you've got at 1,000 viral copies, that even at 100,000 reads, you still get 1x coverage across the entire SARS-CoV-2 30 kb genome. That somewhat drops off when you get down to 110, but here even at 250,000 reads at 10 copies, you can still get 50% of the genome covered at 1x when combining SRSLY with low viral copies and a enrichment panel for SARS-CoV-2. This is what the genome coverage looks like at 1 million read pairs across the SARS-CoV-2 30 kb, 29 kb genome at the different viral copy levels between 1 million down to 10. You can see that you have very high coverage even at 1,000. And then at 1 million, it somewhat drops off, but it's still there for 110. So if you're interested in this further, please look at claretbio.com featured literature. At this app note, we go on to show some other interesting figures where we can use, I'm backing up a slide here, that we can show a deletion as well as some SNP variations in the SARS-CoV-2 genome that we got from the original genome that was sequenced in China. So there are other applications that can be done beyond this analysis to do epidemiological tracking of this. Okay, so those are the two big fields that I cover for SRSLY, the cell-free DNA and the RNA-seq. But I think it's worth briefly mentioning that there are other applications in which SRSLY is good for, and they fall within this kind of Venn diagram of SRSLY's pros, SRSLY's field of use. And so one of them is oligos. This is probably the third most popular use of SRSLY. And by oligos, these are single-stranded synthetic DNA. They can be short, they can be long, they can't be too long because of synthesis machinery and error rates as you synthesize longer and longer. But they're short, they are single-stranded for sure. And if you care about the native termini, the 3' ends, you can do this, but mainly because they're single-stranded. The oligos that people are interested in QCing are sometimes people would like to look at the population density or the population distribution of microarray probes or the fidelity, the completeness of your FISH probes if you're doing kind of histology and stuff. So this is a figure from our paper showing that basically you can look at the percent correct complete length of a 6D-mer versus standard desalting and HPLC. No surprise that with HPLC purification, you get a higher percentage of complete molecules, but you still get a lower proportion of truncations, but they're still present. You can do this and kind of look at, this is just one analysis for this. You can look at where the deletions are, if there's mistakes in there as well. You can use the UMI for this to look at diversity because oligos are kind of low complexity by nature. You can get the complexity higher if you're doing dense microarrays, but if you're looking at individual probes or a pool of probes that are short, UMIs also work very nicely. Ancient DNA. SRSLY technology actually came out of an ancient DNA lab at UC Santa Cruz, and several other ancient DNA labs were the first ones to publish single-stranded library prep methods that they work better for ancient DNA. So as an example of this, what you have is fragment length distribution from rootless hair. Rootless hair is notorious because it doesn't have a root, so it doesn't have a lot of DNA in it, and the DNA that's in it is really highly fragmented. And so if you look at we're able to make very nice libraries from both modern human rootless hair as well as ancient mammoth and ancient woolly rhino. If you go to our literature page and look at the app note for ancient DNA, we go on to show that we can reconstruct the mitochondrial genome of these mammoths and woolly rhinos and do some phylogeny analysis for these. Some other applications that I'm not going to touch on for lack of time is FFPE is another great application for SRSLY. FFPE is notoriously damaged, so therefore it has a higher proportion of single-stranded and nicked DNA than traditional standard high-quality genomic DNA that you'd extract from a fresh frozen sample. Bisulfite treated DNA, bisulfite treatment is by nature very harsh on DNA. It converts, it converts cytosines to uracils, leaves methyl cytosines intact, but it also causes deamination, it causes abasic sites, and it causes fragmentation and causes the DNA to become single-stranded. So SRSLY works very well for bisulfite treatment. There are papers out there showing the benefits of single-stranded library preps for bisulfite. So to get into kind of really quickly just the products for SRSLY, there are two kits that we have, two different chemistries. There's the Pico Plus and Nano Plus. They differ, they overlap slightly in their input range, but basically the Pico Plus has been attenuated, attuned, and perfected for short and naturally fragmented DNA. And I'm talking about cell-free DNA, ancient DNA, kind of forensic samples, short oligonucleotides, whereas the Nano Plus kit is used for higher quality DNA. I don't want to say high quality DNA because we put FFPE in here, but when you compare FFPE DNA extracts to an ancient DNA extract, FFPE is higher quality and more contiguous than ancient DNA. So FFPE, and then it gets better from there. Genomic DNA, cDNA, bisulfite treated stuff. This is generally longer than the ancient DNA and the cell-free DNA, which is why it's put here. And the Nano Plus chemistry allows you to get way more aggressive with your cleans and not flirt that zone between adapter dimer and template insert. So that's what we have. We now offer two kit types, the 24 reaction and the 96 reaction. It's kind of a piecemeal. You can build your own if you want. You can order these with or without primers. You can order the primers as unique dual primers, or you can order the UMI primers. You can order with or without beads if you want. And we supply our own beads, but we also have app notes showing that both the Beckman Coulter Ampere beads and Zymo beads work really well with our samples. I think it's worth noting the price because price is good to be transparent. It drives me personally crazy when I go to a website and they're like, contact us for pricing. So to have the pricing on the website is nice. We have the pricing on our SRSLY quote page, but it's about $25 per reaction without beads and indexes, and $35 if you order more, making us competitive, I would say, with double-stranded preps that you would buy off the shelf. I also want to mention this additional resource of the claretbio.com/featured literature, and this has all of the app notes that as well as the manuscript, which I presented data from, as well as more, if you're interested in more. It's got our posters from all the conferences that we've attended in the past. So with that, I am done at 10:53, which is pretty good timing. So Jessica, Dr. Garcia, has been answering questions online, but I'm gonna just see if there's any questions that are that I can help with or answer or clarify any points or answer any further questions that people have here before time's up. because I haven't been monitoring the questions. I've been doing the presentation. Jessica, do you have any questions?
We've had a couple questions rolling in here at the end that maybe you want to answer. So the first being, what about long single-strand DNA applications?
Sure, let me take that. So long single-stranded DNA, the only SRSLY is agnostic in terms of the length that it ligates to. The major concerns with longer molecules that you have is due to the constraints of an Illumina machine. Illumina machines like DNA to be 600 base pairs or less, maybe 800 base pairs or less, depending on how far you want to push it. Anything above that in bridge amplification won't work on the machine. So that is a function of Illumina. Right, there are ways to get around it. One way to get around it would be PacBio. We currently don't have the ability, the adapters to do PacBio. It is, trivial scientific wise to do it, but in practice, it would take a longer, it would take some R&D in order to develop those adapters. It would take time, but SRSLY will work up to any single-stranded DNA length that'll fit on a Illumina sequencing machine. Anything above that, you will have to shear it. Single-stranded DNA shears very well acoustically with a Covaris or a diagenode bioruptor.
And then the second question, and I will attach a link because it pertains to something that we have as an app note, but maybe you can also expand on it. They ask, does SRSLY perform well after target enrichment?
Yes, it does. So the two target enrichments that I showed in this presentation are exome panel for human biology applications, and that's an exome panel we did with Twist. And then we showed a SARS-CoV-2 enrichment panel that we did with Arbor Bioscience that showed great enrichment for the SARS-CoV-2 genome. The important thing to know is that at the end, after index PCR, a SRSLY library looks just like any other library prep, and it functions just like any other library prep would with downstream enrichment after that. In that the downstream enrichment, they're usually single-stranded probes that are RNA or DNA based, and the DNA is denatured, and then it binds to the probe, and then there's a biotin pull-down step. And SRSLY works just as well as any other prep, as just, we'll attach our app notes to those with the exome panel. We actually compare it to a double-stranded prep as well, so you can see kind of how SRSLY fares for that. And in our upcoming app note, which is using the UMIs in conjunction with a smaller panel to look at enrichment for very low frequency variants, we'll also include a double-stranded comparison there.
Okay, and one last one rolled in at the last minute. Let's do this as well. Can adapter dimers be formed when using SRSLY library prep? And if it does, how can we avoid it when entering into our final library prep?
Yes, yes, SRSLY dimers do form. I didn't go into it in too much detail. I was a little shaky at the beginning of the presentation, and I apologize, but the splint part of the adapters are blocked on both the 5' and 3' end and can't participate in any way into library prep other than Watson-Crick base pairing with the template, which is great. The adapter portion, which is the top portion of the splint adapters, has got a block. The P5's got a block on the 5', the P7's got a block on the 3'. They don't have blocks on what you would need to make a proper ligated molecule. This has the side effect of being able to ligate to each other. The P5 can ligate to the P7 and form dimers. Dimers do happen in SRSLY libraries. The best way to combat that right now we have is with the different SRSLY purification stringencies that we lay out in our kit for your template DNA of choice. By default, you will get more dimers in shorter template molecule types. Ancient DNA and cell-free DNA, where you're going for those short fragments, it's very hard to separate dimers from mappable DNA in that 30 to 50 base pair range. It's just the bead purification is not a scalpel. It's more of a broad brush stroke. So that is one downside. As you get with higher and higher molecular length DNA, such as cell-free DNA, where you're not interested in low fragments, RNA-seq, FFP, bisulfite, oligos that are longer, you run into very little dimer issues. It is important to know which kit, because the different kits, the Pico and the Nano, have different amounts of dimers for different input types. The Pico has less dimers overall for short stuff, and the Nano's got less dimers. They are there. There are mitigation strategies. If you do have high dimers in your libraries, these have been, the protocol and the kits have been set up to minimize dimers, and ideally the libraries normally have no dimers in them. They're not an issue. There are mitigation steps if you do have dimers, and we're constantly working on ways in which to reduce dimers further and to make this way more bulletproof. We good?
Yep.
Okay. Well, everybody, thank you so much for your time, and thank you for joining us for a SRSLY webinar. I will distill this presentation and we'll put it on our website as well, so that if you have colleagues or friends or people that are interested in any aspects of this, please direct them to our website in the near future when this will be up. If anybody has any further questions, technical support at claretbio.com. I monitor that email constantly and am happy to answer any questions and probably send out this presentation to anybody who's interested in it. So thank you so much for your time, and have a good rest of your day.